Have you ever heard of food preservation? So what is exactly the preservation? Preservation is the act of keeping something the same or of preventing it from being damaged. While food preservation is the process of treating and handling food to stop or slow down food spoilage, loss of quality, edibility, or nutritional values, thus allowing for longer food storage. Food can be spoiled by the microorganism if not preserved. Food spoilage is defined as any sensory changes of a food which causes the food to be unfit for human consumption. There are lots of techniques and methods to preserve food. However, in this video, we are going to focus on the use of antibiotics as food preservative. Antibiotics are any of a variety of substances usually obtained from microorganisms that inhibit the growth or destroy certain other microorganisms. The use of antibiotics has been practiced for a very long period of time. It began in 1943, where Tarr and associates already began to explore the use of various antibiotics for the preservation of fish by the use of 2-tetracyclines. While in 1945, Curran and Evans also found that penicillin had a sporicidal effect on a number of bacteria, including Bacillus thermophilus, one of the causes of flat sour spoilage in canned vegetables. In 1955, the Food and Drug Administration permitted the first use of antibiotics in food by the use of chlortetracycline in fresh-dressed poultry. Let's move on to the importance of food preservation. Firstly, food preservation helps in inhibit or destroy the growth of many food spoilage organisms. Food preservation also adds sufficient time to the keeping quality of some foods to permit more efficient and less wasteful distribution of the food. Next, food preservation can prevent, treat, and control the food animal diseases caused by pathogens such as E. coli, Staphylococcus aureus, and Streptococcus pneumonia. Food preservation is also used to increase shelf life of the food product. There are several types of antibiotics used in food preservation. We are going to focus on the common ones, which are tylosin, chloromycetin, and natamycin. Tylosin. Tylosin is a macrolide antibiotic produced by a strain of Streptomyces fredei that has bacteriostatic effect. It is a veterinary medicine that treats bacterial infection in various species. Tylosin is available in the form of injectable, intramammary, and oral formulations. Tylosin is a mixture of 4 components: tylosin A, B, C, and D, in which tylosin A is a major component of tylosin, while tylosin B, C, and D contribute to the overall potency. One of the advantages of tylosin is it is active against most gram-positive bacteria such as Staphylococcus, Streptococcus, Listeria, Enterococcus, Corynebacterium, and Clostridium, Mycoplasma, and certain gram-negative bacteria. Tylosin can control the mycoplasmosis and increase anti-tumor activity of splenocytes in poultry. It can also treat bacterial infections and reduce replications of bacteria. Commonly, tylosin is used for growth promoter in food-producing animals and to lengthen storage period for poultry. Let's see how tylosin works. Tylosin inhibits bacteria by binding to the 50S ribosomes, thus inhibit protein synthesis, which stops bacteria from reproducing but not necessarily kills them. Another antibiotic used as food preservative is natamycin. Natamycin is an antibiotic that belongs to the group of polymicrolides produced by the actinomycetes Streptomyces natalensis. The antibiotic controls the growth of yeast and molds on the surface of cheese and other non-sterile products such as meat and sausages. One of the benefits of natamycin is it is an alternative to traditional chemical preservatives as it meets the demand of natural products. Besides, it is also effective against fungi, unlike sorbates, which slow their growth. A Another benefit of natamycin is it is not dependent on pH level in order to be effective. Natamycin is also effective even at low doses. Furthermore, it does not alter the taste of the food. Another benefit of natamycin is it does not affect the natural maturing process of food as it has no bacterial action. Commonly, natamycin is applied to the surface of dry and fermented sausages to prevent mold growth on the casing. It is also used in cream, cheeses, cottage cheeses, yogurt, and other milk products. Natamycin often used to replace sorbic acid. So how does natamycin work? Natamycin inhibits the growth of fungi by specifically binding to ergosterol present in fungal cell membranes. The molecule interacts irreversibly with the sterol, forming a sandwich-like structure embedded in the hydrophobic core of the membrane between the two lipid layers. This causes membrane fragmentation and indirectly affects the function of ergosterol. Now we are going to understand about chloromycetin, also known as chloramphenicol. Chloromycetin is an antibiotic first isolated from cultures of Streptomyces venezuela in 1947 but currently produced synthetically. Chloramphenicol can be obtained from the filtrate of the Streptomyces venezuela culture by extraction with alkyl acetate. It has been evaluated as genotoxic, which could cause genetic disease, possibly lead to cancer. Besides, it is also known to cause an extremely serious disease called aplastic anemia. Chloromycetin is classified as food additive, which may not be used in any form of food-producing animals. Therefore, chloromycetin are not allowed for food animal production and aquacultures in Malaysia. Advantages of chloromycetin. Chloromycetin slows the growth of bacteria by preventing them from producing important proteins that they need to survive. It also plays a role as an antimicrobial agent. Chloromycetin is also a potential cytotoxic agent effective against myeloma cells. Furthermore, it has an excellent activity against anaerobes. Chloromycetin is effective in suppressing the growth of undesirable microorganisms. Chloromycetin is widely known for its use in milk preservation. It also used to lengthen the storage time of raw food, especially meats, fish, and poultry at chilling temperatures. Now let's see how chloromycetin works in details. Chloramphenicol is a protein synthesis inhibitor in bacteria. It inhibits protein by binding irreversibly to the bacterial 50S ribosome subunit. Chloramphenicol hinders the access of amino acid tRNA to the A site for amino acid incorporation by acting as a peptide analog. It prevents formation of peptide bonds. Thus, it inhibits protein synthesis at the peptide transferase reaction. And that's all from us. Hope you are enjoying the video. Feel free to ask a question if you have any doubt. Thank you and have a nice day.